Okay, so post-transcriptional control. We're going to talk a little bit about transcription in prokaryotes first, and then we'll compare it to eukaryotic transcription. So in bacteria, all transcription occurs in an RNA polymerase enzyme complex consisting of 5 subunits. So bacteria, humans have a couple of different RNA polymerases. Bacteria just has this one complex. And like I said, it consists of 5 subunits. I don't think it's super important to know all of the just to know all the names of the subunits, but I would say it is important to understand what the sigma factor is. And that's basically an additional subunit that this complex has to associate with before it becomes active. So once that sigma factor associates with the RNA polymerase complex, it becomes active and it can start polymerizing. So transcription consists of 3 phases: initiation, elongation, and termination. So we'll just go over each of them individually. The first, obviously, is initiation. And during initiation, RNA polymerase scans the prokaryotic DNA until it finds a promoter sequence. So recall that a promoter is a DNA sequence at which an enzyme can attach to initiate transcription. So that's just the starting point, pretty much, for RNA polymerase to attach and then begin transcription. So in bacteria, there are 2 primary sequences found in promoters. And that's really what promoters are. They're just specific sequences that allow these enzymes to attach to the DNA so they could start transcription. So those 2 primary sequences are known as the Privno box and the 35 sequence. Again, the name's not super important, but you should definitely just understand what a promoter is and why it's important. So once the promoter is located, the enzyme and the DNA form what is known as a closed complex. RNA polymerase then unwinds and separates the small portion of the double-stranded DNA. This single-stranded sequence and the enzyme together are known as the open complex. So once we switch from the closed complex to the open complex, we're going to start to move towards elongation, which is our second step. So after the open complex is formed, elongation begins and the sigma factor dissociates from the RNA polymerase complex. As its primary functions are to increase the enzyme's ability to recognize promoters and to decrease the non-specific affinity for DNA of the enzyme. So the sigma factor that we were talking about on the last slide is super important for RNA polymerase to find the promoter, but once it finds that promoter, the sigma factor dissociates and we don't really need it anymore. So RNA polymerase moves down the bacterial DNA in what is known as a transcription bubble. And that's kind of what you're seeing right over here. This isn't the greatest graphic to see the transcription bubble, but it's evident enough right here. It's called a bubble because it's just a small section where the DNA is unwound and separated, so it appears like it's a bubble. And that's how RNA polymerase moves down the strand of DNA. So one polymerase complex will synthesize the entire RNA molecule. Okay, and then finally termination. So after elongation, termination begins when the complex receives a termination signal, often with the help of a protein row. That's really all you need to know about the protein row. It's just a termination signal that aids in the switch from elongation to termination in prokaryotes. So when the signal is received, the complex falls off the DNA and releases the newly synthesized RNA molecule. And that transcription bubble on the DNA closes and the RNA polymerase will repeat the cycle, again, starting with the association of the complex with another sigma factor. Because remember, it's an enzyme, so it's not used up in the reaction, but it does need to find another sigma factor before it can find that promoter and start transcribing again. And we're going to switch over and start talking about eukaryotic transcription and modification. So as we did mention before, eukaryotic transcription takes place in the nucleus. So remember, prokaryotes don't have a nucleus. That's why they're prokaryotic. So transcription and translation can occur simultaneously. That does not happen in eukaryotes. Our transcription is in the nucleus and translation occurs in the cytoplasm. So during transcription, eukaryotes produce an mRNA precursor known as hnRNA. This hnRNA will be extensively modified in the nucleus before leaving as mRNA. So that's another difference between eukaryotes and prokaryotes, is that immediately following prokaryotic transcription, that molecule is a mature mRNA molecule and it is completely ready for translation. When we're talking about eukaryotes, it's a little bit different of a story. The primary transcript is hnRNA, which is just a very long version of mRNA that needs to be cut, is pretty much what it is. It just needs to be cut and modified before it leaves. So the hrNA contains many non-coding sequences, which are known as introns. And between those, there are sequences that actually do code for the proteins, and those are known as exons. And the RNA strand will undergo splicing in the nucleus. So the spliceosome is a complex made of proteins and catalytic RNA molecules known as snRNA molecules. So spliceosomes are not pre-assembled, but assemble spontaneously around the individual intron that needs to be removed. So intron, we said, is that not coding, that non-coding those sequences. So the spliceosomes actually form around those introns and remove them. So that's part of the alternative splicing that takes hnRNA and turns it into a mature mRNA. So this post-transcriptional RNA modification is so powerful because a single hnRNA can be spliced into multiple different ways. And that creates a different final mRNA products and eventually different protein products. So that's something that's that eukaryotes do, prokaryotes don't do it. This is known as alternative splicing. So we can have multiple protein products coming from the same gene. So that does make it a very powerful tool, and it makes it really efficient because we can get much more protein products, but we don't have to increase the amount of mRNA or the amount of genes that we have to express. So more modification, once the RNA molecule has been spliced, it undergoes further modification before leaving the nucleus. The hnRNA will acquire a methylated guanine nucleotide on its 5' end, and that's known as the 5' cap. It's either known as the 5' cap, sometimes I've heard it called a guanine cap. And on the other side, it's also going to acquire a string of several hundred adenine nucleotides on its 3' end, and that's known as the 3' poly A tail, or simply the poly A tail. So both the cap and tail are essential for preventing the mRNA's degradation by exonucleases when it leaves the nucleus for translation. So it leaves, there are these exonucleases that are kind of floating around in the cytoplasm, and their job is to degrade RNA. So this 3' poly A tail and the 5' guanine cap actually prevents these exonucleases from destroying them before they can get to translation. So those are super important because without them, we wouldn't have protein production. So which of the following is true regarding post-transcriptional modification in prokaryotes? Take a minute, pause your video, and we will go over it. Okay, which of the following is true regarding post-transcriptional modification in prokaryotes? So the correct answer is C. The sigma factor associated with RNA polymerase will detach during elongation. So let's go over it. A, the hRNA produced must be extensively modified before it can be translated into proteins. So that is true for eukaryotes. So you have to remember that prokaryotes, the primary transcript after transcription, there's mRNA. They don't do alternative splicing. They don't have this immature form of RNA that eukaryotes do called hnRNA. So if we were talking about eukaryotes, that would definitely be true, but prokaryotes, it does not occur. Spliceosomes are assembled spontaneously around the intron they are removing. Again, that would definitely be true for eukaryotes, but we don't see alternative splicing in prokaryotes, so there is no spliceosome. So B would also be incorrect. D says RNA polymerase in prokaryotes moves down the DNA in what is known as a replication fork. It doesn't. It moves down in what is known as a transcription bubble. Replication fork would obviously be for replication. So the only true answer is going to be C. The sigma factor associated with RNA polymerase will detach during elongation, which is true, like we just spoke about. Remember, when it starts elongation, it does not need the sigma factor anymore. So the sigma factor dissociates, and it won't associate with it again until it needs to attach to a new DNA molecule.